Well, welcome to Click for IB Biology. We're studying genetics. We're looking at monohybrid inheritance patterns. We're doing two genes with two alleles, but in this case, we're looking at codominance. There's a blank version of this presentation listed below in the description. And of course, we have a quiz with its 10 questions and answers that you can use to consolidate your understanding of this topic. So codominance, monohybrid cross. So we follow the same pattern. We're going to write out our parental phenotype, then our parental genotype. We will use the Punnett square to work out the offspring genotypes. We will then record those and their ratio, and then we will state the phenotypic ratio at the end of the first generation. The topic that we're going to choose is the MN blood group, in which glycophorin A takes two forms. It's M or N, which makes this very simple to add an allele key. So since it's codominance, we can use different letters. If you do dominance, you have to use the same letter in the upper and lower case. So the parental phenotype that I would like to use is that we have someone who's in blood group M. Oh, sorry, there's at one point M and N are codominant. They both influence the phenotype. So I'd like to start with blood group M, so that's the blood group, and blood group N is the blood group. Now this means that our genotypes will be MM at the gene loci and NN at the loci. Meiosis will segregate these alleles so that the gametes are big N, big N, big M, big M. And so in the fertilizations which take place, we would end up with the heterozygous big M, little n, big M, big M, N, big M, N, big M, N. And so the offspring of the genotypes from these fertilizations in the center here are all predicted to be big M, N, which is the heterozygote. Now, because we are codominant, the offspring are described that their blood group is the MN blood group. And so this is a third distinct phenotype. Remember, the first phenotype was blood group M, the second phenotype was blood group N, and the third phenotype which has emerged is the heterozygous, which we will call MN. If we then take people who are blood group MN, and what we're going to do, of course, is perform another genetic cross between two heterozygotes, like so, and at this gene loci we would be M, at this loci would be N, and that repeats here since they're genetically the same. And meiosis will segregate the alleles, so half of the gametes will be M, N, half the gametes will be M and N. So then, sorry, apologies, those should be in the boxes. So then if we have fertilization between this sperm cell and this egg cell, we would return to the homozygous dominant MM. Then we would have MN, we would have MN there, and then of course NN. And so our genotypes of our offspring, 1/4, a probability of 1 in 4 of MM. Then there would be 2 of MN, as you can see, this one here and this one here. And then finally, 1/4 of the offspring probability is that they are NN, like so. Now, when we come to the offspring phenotypes, then we will end up with 3 distinct phenotypes, because this ends up as blood group M, this is blood group N, and then we would end up with 2 MN. So let's just go through the probability of that then. So 1/4, 1 in 4 chance of blood group M from a heterozygous cross. There's a 0.5 or 50% probability of MN, the third phenotype, and we back to 1/4 or 0.25 of N, like that. So codominance does make a difference in the outcome of the F2 ratio. Okay, so we're going to carry on looking at monohybrid crosses. Another variation is coming up in the next video. There's the playlist, the subscription. You can click that for the channel. There's the playlist for all of our genetics videos.